Related. Today I'll talk about the basic of protein folding. This presentation is supervised by Professor Usman Sumofren Tambunan. Now let's talk about the protein first. Protein is a macromolecule that composed from many amino acids, usually more than 50. Unlike the other macromolecules, protein adopt a specific 3D conformation. The specific 3D conformation is able to fulfill a specific biological function. Protein structure, often called as native fold. The native fold has a large number of favorable interactions within the protein. There is an entropy cost to folding protein into one specific native fold. As we know, the protein structure is divided into 4 groups: the primary, secondary, tertiary, and quaternary. The primary structure of protein is defined as the sequence of amino acids linked together by the peptide bond to form a polypeptide chain. Secondary structure is comprised of regions stabilized by hydrogen bonds between atoms in the polypeptide backbone. Tertiary structure is the three-dimensional shape of protein determined by regions stabilized by interaction between the side chain. The quaternary structure of protein is the association of several protein chains or subunits into a closely packed arrangement. Each of the subunits has its own primary, secondary, and tertiary structure. The subunits are held together by hydrogen bonds and van der Waals forces between nonpolar side chains. As we can see here at the top is the peptide bond. Each peptide bond has some double bond character due to resonance and cannot rotate. There are 3 bonds, separate sequential alpha carbons in a polypeptide chain, is what we can see on the bottom. The NC alpha and C alpha C bonds can rotate, described by dihedral angles designated by C and P, respectively. The peptide CN bond is not too free to rotate. Other single bonds in the backbone may also be rotationally hindered depending on the size and charge of the R groups. This is the main secondary structure. There are one more named as the random coil. The primary structure consists of a sequence of amino acids linked together by peptide bonds. The resulting polypeptide can arrange into units of secondary structure, such as helix in this picture. The helix is a part of the tertiary structure of the folded polypeptide, which is itself one of the subunits that make up the quaternary structure of the multi-subunit protein, in this case, hemoglobin. And now let's talk about the protein stability. Native protein are only stable under physiological condition. Native structure results from a delicate balance of many forces such as hydrogen bond, covalent interaction, non-covalent interaction, etc. Protein are stabilized by several forces. First is hydrophobic effect. Protein structure mainly governed by the hydrophobic effects that causes protein minimizes their contact with water. This could reduce the entropy value. And second is electrostatic interaction. Relatively weak van der Waals forces are important to stabilize native structure. The strong H bond makes only a minor contribution to stabilize the native structure. Third is metal ions. Metal ions that bound within a protein could contribute to the protein stability. Generally, metal ions stabilize the small domain of protein. For example, the zinc fingers are motifs that frequently occur in DNA binding proteins. Proteins are Dynamic structures. Proteins are flexible and rapidly fluctuating molecules. The mobility is important for protein function. Proteins can undergo denaturation and renaturation. Now let's talk about the protein denaturation. Low conformational stability of native proteins makes them susceptible to denaturation. Protein can be denaturated by heat, pH, detergents, and chaotropic agents. Heat could melt the structure, pH could alters ionization state of the side chains, detergents breaks the hydrophobic interactions, and chaotropic agents disrupts the hydrophobic interactions. At the right side, we can see the graph of ribonuclease denaturation at different temperature and different GD and HCl concentration. Let's talk about the denaturation and renaturation experiment. This experiment was firstly conducted by Christian B. and Vincent by using ribonuclease A. The protein was denaturated by using egg-molar urea and renaturated by dialysis with full enzymatic activity, as what we can see in this slide. This slide is containing the procedure to denaturate and renaturate the enzyme. Now let's talk about the protein folding patterns. Two terms are used to describe a folding pattern is motif or fold and domain. A motif is a recognizable folding pattern involving two or more elements of secondary structure and the connection between them. Motifs can be simple and can be also elaborate. In some cases, a single large motif may comprise the entire protein. The term fold is much more commonly applied to describe more complex protein folding. For example, the distinctive arrangement of 8 alpha helices in myoglobin is found in all globins and is called the globin fold. Larger motifs can be constructed from smaller motifs. As this slide, we can see at the bottom a series of beta-alpha-beta loops arranged so the beta strands form a barrel creates a particularly stable and common motif, the alpha-beta barrel. In this structure, each parallel beta segment is connected by its neighbor to an alpha helical segment. And let's talk about the domains. A domain is a part of a polypeptide chain that is independently stable or could undergo movement as a single entity with respect to the entire protein. Each domain can appear as a distinct globular lobe region. The two or more domains of a protein often have different functions, which are coordinated in the overall function of the protein. Small proteins such as myoglobin usually only have one domain. At the right side, we can see the troponin C structure. Now let's talk about the left frontal paradox. First, we assume a chain of 100 amino acids, and then only 3 conformations allow. The possible conformation is about 5 times 10 power of 47. And second, we assume that bond rotation rate is 10 power 12 per second. Time to try all the conformation is roughly about 2 times 10 power 28 years. The conclusion is protein folding is not a random event, but has a specific pathway. This is the summary of today's presentation. Protein consists of more than 50 amino acids with a specific 3D structure. There is 4 levels of protein structure: primary, secondary, and tertiary and quaternary. Protein is stabilized by several forces. Protein folding has patterns, motifs, and domains. Protein folding has a specific pathway. I think that's all for my presentation today. Thank you for the opportunity and stay safe.